In this part, we'll be talking about digestion and absorption along the gastrointestinal tract. We have to consider some points with regard to digestion and absorption. No absorption at the level of the esophagus. We have little at the level of the stomach. The organ where we have the majority of absorption is the small intestine. The small intestine also is highly specialized, having highly specialized structure to increase the process of absorption. We'll see that specification of this organ. Most of nutrients are absorbed before reaching the ileum. Also, we have at the colon final removal of electrolytes and water. What makes the small intestine as the specialized organ? First of all, the mucosa is folded. We have folds of Kerkring, which are circular folds of the mucosa. In addition, we have also villi, which are increasing the surface area. We'll see also the structure of these villi. They are highly vascularized, which is important for fast removal of absorbed nutrients. In addition, also, we have lymphatic vessels, which are important for the absorption of lipids. Over these villi, we have epithelial cells. The apical membrane of these epithelial cells is also folded to give what we are calling microvilli or brush border. That specialized structure is increasing surface area about 600 times. So these are the structure. We have folds over the membrane. Over these folds, the first picture, we have villi. The villi are highly vascularized. In the center of these villi, we have central lacteals, which are the lymphatic vessels, and they are important for absorption of lipids. The membrane of epithelial cells, which are lining villi. The apical membrane of these cells is folded to form what we are calling microvilli or brush border. So as you see, this is the structure of the small intestine. We have villi, highly vascularized. In the center of these villi, we have central lacteals where we have lymphatic circulation at that level. In addition, the wall of the small intestine is having muscular mucosa, which is important for movement of these villi. These villi are waving within the chyme, which is enabling better absorption of nutrients from chyme. In addition, this muscular mucosa is important for changing the position of the folds that we are having over the mucosa, shortening and elongation of these folds. In addition to this, we have also what we are calling brush border enzymes at the level of the mucosa, which are important for the process of digestion. We will see these brush border enzymes and their specification in the process of digestion. After this introduction about digestion, we are starting talking about the digestion of carbohydrates. So we are ingesting carbohydrates in many forms, in the form of starch, which are polymer of glucose ramified at specific position. So these glucose molecules are linked to each other by what we are calling 1,4-alpha bond, but at the ramification, the type of bond is alpha 1,6 linkage. In addition to starch, we are also ingesting carbohydrates in the form of saccharose found in fruits, in the form of lactose, which is found in milk products. Also, we are ingesting carbohydrates in the form of cellulose. But the type of linkage between the cellulose molecules, they are similar to the starch molecule, but the type of linkage between the glucose molecule, we have it in the form of 1,4-beta linkage. The process of digestion begins in oral cavity by the activity of salivary amylase, but after the process of swallowing and during gastric phase, the low pH of the stomach is reducing activity of the salivary amylase. The optimal activity of the amylase is at alkaline pH. So after gastric emptying, we are needing more amylase, and we have the pancreas releasing that enzyme, which can digest about 50 to 80% of starch. So this amylase or pancreatic amylase is attacking the 1,4 linkages, the alpha form of the 1,4 linkages to get maltose, maltotriose, and alpha limit dextrins. In addition, we are continuing the process of digestion at the level of the small intestine by the activity of brush border enzymes. We have more enzymes at that level. We have lactase, we have saccharase enzyme, we have maltase enzyme. Also, we have the alpha dextrinase enzyme, which can attack the 1,6 linkage between glucose molecules at the ramifications. So by the activity of amylase, which is attacking at every two glucose molecules, we are getting maltose. For the digestion of maltose, we have brush border enzyme, which is called maltase. By digestion with maltase, we are getting from each maltose molecule two glucose molecules. In addition, also brush border is having other enzymes for digestion of saccharose, for digestion of lactose. We have saccharase enzyme, the saccharose is also a disaccharide formed of glucose and fructose. By the activity of saccharase, we are getting also glucose and fructose molecules. In addition, we have also lactase enzyme, which can digest lactose as disaccharide to get finally glucose and galactose molecules. At the brush border also we have dextrinase enzyme which can digest the 1,6 alpha bond at ramification. By this digestive process we are getting maltose and glucose. So, after the final digestion of carbohydrates in intestinal lumen and by brush border enzymes, we are getting all carbohydrates in the form of monomers, glucose, fructose, and galactose. So, this slide is showing you brush border enzymes like lactase, maltase, and saccharase. And after having all these foodstuffs in the form of monomers, we have absorption of these foodstuffs with the help of carriers. We have carriers also at the brush border for absorption of glucose. We have carriers for absorption of galactose, and also we have carriers for absorption of fructose. So how we are getting absorption with the help of these carriers. Absorption for glucose is by what we are calling sodium dependent carriers, which means that these carriers are depending on concentration gradient for sodium between the lumen and inside the cells to get what we are calling secondary active co-transport mechanism for absorption of glucose. In addition, glucose also, we have another mechanism for absorption which is called absorption by solvent drug. How this is taking place when we have high concentration of glucose in chyme by getting removal of water through the paracellular space, that diffusion of water is taking with it also glucose molecules through the paracellular space. So this type of absorption is called absorption by solvent drug. For galactose absorption, also we have specific carriers for galactose which are sodium dependent carriers. So the mechanism is also secondary active transport mechanism. While for fructose, we have sodium independent carriers, which means the type of absorption is by facilitated diffusion. So as you see, at the brush border, in addition to enzymes, we have carriers. We have sodium-dependent carriers for transport of glucose from the lumen toward inside the cells. We have also carriers for galactose. Also, they are sodium-dependent carriers. Also, we have carriers for fructose, which are sodium-independent carriers. Once we have these foodstuff absorbed and having them inside the absorptive cells, they are removed toward interstitial fluid by the help of carriers also at the basolateral membrane. But these carriers are transporting them from the high concentration inside the absorptive cells toward the interstitial fluid where they can be found in a lower concentration. Once we have this foodstuff in the interstitial fluid, these are hydrosoluble, so they can be taken away from the eye with the help of blood circulation. You have to note also at the basolateral membranes, we have sodium-potassium pumps. These pumps are important to ensure having lower concentration of sodium inside the absorptive cells This process is important for the activity of the sodium-dependent carrier, like galactose carriers and glucose carriers. So we can summarize the process of digestion and absorption of carbohydrates. We are starting this process by the activity of salivary amylase, then followed by the activity of pancreatic amylase to get starch digested into maltose, maltotriose, and alpha-dextrin. Then these stuff are digested furtherly by the activity of maltase and alpha-dextrinase, which are at the brush border to get final digestion of starch into glucose. Also, we have process of digestion for lactose by the activity of lactase at the brush border. Also, we have a process of digestion for saccharose by the saccharase at the brush border to get finally all carbohydrates in the form of monomer. Then we can absorb these food stuff in that form. So we cannot absorb any of carbohydrate products in the form of dimers, only in the form of monomers. Patients suffering from lactase deficiency, for example, they cannot digest lactose, so the lactose cannot be absorbed and may generate some health troubles for these patients, like intestinal discomfort. Also, they are not tolerating milk or any lactate products. Also, they are developing what we are calling osmotic diarrhea. Then once we have the foodstuff in the form of monomers, they are transported across plasma membranes. Now we'll start talking about digestion and absorption of proteins. We are ingesting also proteins, and we have a process of digestion for proteins. The process of digestion begins in the stomach by the activity of pepsin. You have to note that the activity of pepsin, or the optimal activity of this enzyme, is at acidic pH. Once we have emptied the content into the duodenum, the activity of this enzyme is much reduced by the alkaline pH that is at the lumen of the small intestine. Also, we are needing now more enzymes for the digestion of proteins. We are secreting actually these enzymes from the pancreas. We have enzymes with endopeptidase activity like trypsin and chymotrypsin. Also, we have enzymes with exopeptidase activity like carboxypeptidase. By the activity of these enzymes, we are getting digestion of proteins, but still we will have small peptides, and also we are getting, as a result of digestion, more amino acids. At the brush border, we are continuing the process of digestion by the activity of amino peptidases, which result. By that process of digestion, we are getting smaller peptides and more amino acids. The smaller peptides like dipeptides, tripeptides, tetrapeptides can be absorbed by the luminal membrane of the absorptive cells, and then the last level where we are having digestion process is inside the absorptive cells by the activity of intracellular peptidases. So these dipeptides, tripeptides, tetrapeptides, after the process of absorption, everything we are getting digestion, all proteins in the form of amino acids. So at the brush border, we have also amino peptidases, which can digest small peptides into smaller peptides, which can be absorbed by carriers at the brush border toward inside the absorptive cells where they are undergoing further digestion, which is the last process of digestion by intracellular peptidases. So finally, we are getting all proteins in the form of amino acids. All these amino acids can be removed by carriers at the basolateral membrane toward interstitial fluid. Then they are taken away from villi by blood circulation. So, as process of absorption, we have to deal with the absorption of smaller peptides like dipeptides, tripeptides, tetrapeptides, and these peptides are transported toward enterocytes by sodium-dependent carriers. So the process of transport is secondary active co-transport for these small peptides. As you see from the process of absorption, unlike carbohydrates, small peptides can be absorbed in the form of dimers, trimers, and tetramers, while carbohydrates cannot be absorbed in that form. In addition, we have to deal with the absorption of amino acids. For amino acids, also we have carriers at the border membranes, which can transport these amino acids from the lumen toward inside the absorptive cells. For absorption of amino acids, we have carriers at the luminal membrane. Some of these carriers are characterized as sodium-dependent carriers. We have at least three different types have been characterized. Some of them, of these sodium-dependent carriers, are for neutral amino acids. We have some carriers for proline and hydroxyproline. We have also some carriers for phenylalanine and methionine. In addition to sodium-dependent carriers, we have also sodium-independent carriers for some basic and neutral amino acids. So as summary, at the luminal membrane, we have aminopeptidases, we have carriers that are dealing with transport of small peptides across luminal membrane, and also we have carriers for amino acids which are dealing with the absorption of amino acids. Once we have final digestion by intracellular peptidases, all these amino acids can be removed from the absorptive cell toward interstitial fluid, and they are taken away from the eye by blood circulation. So for lipid digestion and absorption, we have to consider that we have very little or no digestion or absorption of fat in the stomach. At the level of the small intestine or duodenum, we have a process of emulsification of lipids. So big droplets of lipids are transformed into smaller droplets of lipids by the activity of bile salts. This is showing you the function of bile salts. If we have no bile salts, lipids in chyme are forming large droplets of lipids. But by the presence of bile salts, these large droplets of lipids becoming smaller and smaller droplets to get finally formation of what we are calling micelle and the lumen of the small intestine. As you know, the bile salts are amphipathic molecules, so they are having lipophilic part and hydrophilic part, and by orienting themselves, the hydrophilic part, which can give orientation of these bile salts toward the aqueous media, while the lipophilic part, which is their orientation toward the fat media in these droplets. The process is important for the digestion and absorption of lipids. In that way, we are getting an increase in the surface area for the attack of enzymes. As you see, the large droplets of lipids as having much lower surface area than the sum of all smaller droplets formed or micelles formed, and these micelles which are formed are having much higher surface area. It is important for the attack of enzymes. So this is the structure of micelle. As you see, it's having hydrophobic core and hydrophilic shell. And that's the orientation of lipid molecules and bile salts in micelle structure. As you see, glycerol part of the triglycerides is oriented toward the hydrophilic shell, and also the hydrophilic part of bile salt is oriented toward the hydrophilic shell, while toward the hydrophobic core we have orientation of the hydrophobic part of bile salts and also the orientation of fatty acids of these triglycerides are orienting themselves toward the hydrophobic core. That's important for the activity of enzymes. You know, the enzymes are hydrosoluble, so they cannot go inside that big drop of lipids to digest lipid molecules. It's attacking at what we are calling oil-water interface at the surface of the hydrophilic shell, so they can start digestion of these triglycerides at that level, at oil-water interface between the hydrophobic part and hydrophilic shell. By the activity of lipolytic enzymes, triglycerides are digested into monoglycerides, and we are getting free fatty acids released. Then we have a process of absorption for these stuff from the lumen toward inside the absorptive cell. These are liposoluble, so they can be transported across the luminal membrane simply by simple diffusion. The membrane also a lipid structure. These are lipid particles, so they can be transported across plasma membranes of absorptive cells by simple diffusion. Once we have them inside the absorptive cells, these monoglycerides and free fatty acids are reforming back triglycerides. Then triglycerides with cholesterol, phospholipids. Also, we have a protein structure which is formed by the absorptive cells called B-lipoproteins are conjugated together or combined together to give the formation of kilomicrons, which are having around 60 to 750 nanometer diameter, and these kilomicrons are expelled out of the absorptive cells by the process of exocytosis. Once we have digested lipids at micelles, and once we have micelles becoming in contact with the luminal membrane, we are having simple diffusion of fatty acids and monoglycerides and also modified cholesterol molecules toward inside the absorptive cells. Now, inside the absorptive cells, monoglycerides and fatty acids are reforming back triglycerides. Why we have needed the process of digestion? We are reforming these triglycerides, we are reforming back triglycerides. Actually, the process of absorption of these smaller molecules like monoglycerides and fatty acids is much easier than the absorption or the transport of triglycerides across the plasma membrane. Inside the absorptive cells, we are forming that aggregates of lipids with the B-lipoproteins, which is called kilomicrons, and then kilomicrons are expelled out at the basolateral membrane towards interstitial fluid by exocytosis. These kilomicrons are taken away from villi by lymphatic circulation. So we have lymphatic vessels at the villi, which are called central lacteals, and these central lacteals are collecting kilomicrons, and we have the lymph collected and reintroduced in blood slowly through the thoracic duct. So we can summarize that we have pancreatic lipolytic enzymes which are digesting big lipid molecules to get the smaller lipid molecules at the level of micelles inside the lumen of the small intestine. So these smaller lipid molecules can diffuse across the plasma membrane of the absorptive cells toward inside the absorptive cells. Any pathological condition which results in decreasing digestion and or absorption process of these lipids results in increasing fat content in stool, which gives the whitish or yellowish discoloration of the fecal material. This medical condition is called steatorrhea which means increased fat content in stool. This part we'll be talking about absorption of water and electrolytes. So we'll start with the absorption of sodium. At the basolateral membranes, we have active transport for sodium by the activity of the sodium-potassium pump. So by transporting of sodium from inside the absorptive cell towards interstitial fluid, we are keeping concentration gradient for sodium between the lumen and inside the absorptive cell, which is important for co-transport mechanism of other molecules like glucose, like galactose, like small peptides, and amino acids. Once we have transported sodium towards interstitial fluid, we are creating a potential across the whole absorptive cell between the interstitial fluid and the lumen, which is transcellular potential. More positive toward the interstitial fluid, which is attracting negatively charged particles from the lumen of the small intestine, and we have plenty of chloride which can be absorbed in that way. So the absorption of chloride is driven by the electrical gradient, which is established by the absorption of sodium. Once we have absorbed sodium and chloride, we are increasing the osmotic pressure at the interstitial fluid. The increased osmotic pressure will increase osmosis of water from the lumen toward interstitial fluid. So we have also more water by this process. For the absorption of potassium, potassium is actually moving across membranes according to the electrochemical equilibrium. It's absorbed passively in the small intestine, while in the colon we have a process of secretion actually of that potassium in exchange with sodium. We have a special problem with the absorption of calcium. Calcium is transported actively. How the process of transport is taking place, we have special mechanism for that. Enterocytes are forming a protein molecule to which we can have calcium binding. These protein molecules are called cal binding. Cal binding are transporting calcium across the cell, the absorptive cells. At the basolateral membrane, we have pumps for calcium, so we have the process is active process. We have calcium pumps at that level, which can remove calcium from inside the absorptive cell toward interstitial fluid. But I think you know calcium absorption can increase by vitamin D and parathyroid hormone. How the process of calcium absorption can be regulated at that level. So we said we are having cal binding, to which we can have calcium binding and transported across the absorptive cells toward the basolateral membranes where we have the pump. Now, the regulation of calcium absorption is achieved by increasing expression of cal-binding inside the absorptive cells. Once you have increased cal-binding concentration by the activity of parathyroid hormones or vitamin D, you are getting much lower concentration of free calcium inside the absorptive cell, and you are ensuring more calcium ions to move from the lumen toward inside the absorptive cells. So in that way, we are getting the regulation of calcium absorption by increasing expression of cal-binding inside the absorptive cells. We have also a special problem for the absorption of iron. First of all, iron is absorbed in the ferrous form, Fe++, than the ferric form. Secondly, we have also some constituents in there can enhance iron absorption. Also, we have some other constituents that can reduce absorption of iron. For example, vitamin C, the acidic content of the stomach can increase iron absorption, while phosphates, oxalates, phytic acids, which are found in cereals or alkaline media, can inhibit iron absorption, probably by getting the ferric form of iron transformed into the ferrous form or vice versa. That enhancement or inhibition of iron absorption. The process of iron absorption is also active mechanism. The most acceptable theory for iron absorption, we have the absorptive cells forming a protein structure which is called apoferritin. That apoferritin is released first in the lumen of the small intestine. Then the apoferritin is binding to the iron. By binding of apoferritin to iron, we are forming ferritin molecules. At the luminal membrane of absorptive cells, we have receptors for the ferritin molecules. So, by binding of ferritin to their receptors, we are activating a process of endocytosis. So, the whole process of iron absorption is receptor-mediated endocytosis. Now, that iron remains at the level of the absorptive cells and can be transported toward body fluids if there is a need for iron. If there is no need for iron, iron remains at the level of the absorptive cells. This process is called mucosal block, which means we are blocking the absorption of iron at the level of absorptive cells. Now, how your body knows that there is a need for iron or there is no need for iron? You know, in our body fluids and the blood, iron is transported by another protein structure which is called transferrin. Now, if you have all transferrin molecules loaded with iron, that means there is no need for iron to be moved from the absorptive cell toward interstitial fluid. But if there is an increase in the concentration of unloaded transferring molecule with iron. That means there was some consumption for this iron, and there is a need for this iron, so we can have that iron move from inside the absorptive cell toward interstitial fluid. Now, the iron absorbed and stored at the level of absorptive cells can be lost. You know, these cells are disconnecting every 3-4 days. We are losing these cells, so we are losing also iron, which is stored in these cells. This phenomenon, again, it is called the mucosal block, which means we are blocking the absorption of iron at the level of spinal mucosa. If we are needing that iron, we are taking it. If we are not needing that iron, we are losing it by the process of disconnection of these cells. In this part, we are going to talk about the absorption of vitamins. Most vitamins are absorbed in the upper part of the small intestine, but we have also some vitamins like vitamin B12 is absorbed in the lower part of the small intestine. You know, we have water-soluble vitamins and we have lipid-soluble vitamins. With regard to water-soluble vitamins, most of them, they are absorbed passively. We have also some vitamins which can be absorbed actively, like vitamin C, vitamin B1, vitamin B12. You have to note also that for the absorption of vitamin B12, we are needing intrinsic factor, which is released from the oxyntic cells of the stomach. Lipid-soluble vitamins like vitamin A, D, E, and K, they are absorbed in the same way as lipids. So first they are solubilized in my cell, then transported across the luminal membrane of the absorptive cells by simple diffusion, then incorporated in kilomicrons, then they are taken away from the villi with the help of lymphatic circulation. You have to note one thing with regard to the absorption of these vitamins. Any medical condition which results in maldigestion and or malabsorption of lipids may result in decreased absorption of one or more of these vitamins and generating medical conditions related to the deficiency of one or more of these vitamins.